Okay, this is the first video for the fifth chapter of this course. And the fifth chapter, chapter 5, is going to be about percolation and growth. Okay, it's about the spreading of materials or a phenomenon through heterogeneous spaces. Okay, so yeah, let's get started. So spread processes, they are actually everywhere in the real world. Okay, the definition is the wider distribution of some property. This property could be particles, individuals. These individuals can be humans, animals, um, or any agent. Okay, over space with respect to time. With respect to time. Some examples here: wildfires, right? Invasive species, infectious diseases, epidemics, and even innovations, gossips, information, and urbanization. They all can be considered as properties, and they expand. They move across space. They spread across the space. Okay, which means that using percolation and growth models, you can simulate this phenomena. Okay, these examples here. And there are two types of spread models. The first type is called percolation models, or we can see that they're based on percolation theory. And this type of models focuses on the nature of the media through which spread is occurring. For example, a contamination is spreading in soil, and soil is the media of the spread. Okay, the property of the soil, the physical property, chemical property, and the structure, spatial structure of the soil affects the spread of contamination. For percolation models, we focus more on the media, on the soil itself, instead of the spreading process of this contamination. If you're focusing on the spreading process of this contamination, you can use growth models, which is the second category of spread models. Growth models, they focus on the dynamics of the spread process, which means the media is not that important. We focus on the spread process of this contamination. Okay, so in this chapter, we're going to discuss both of them. And we will start with percolation theory or percolation models. Okay, what is percolation? So movement of material in heterogeneous media. What is a heterogeneous media? It means that the structure of the media, the property of the media, they do not distribute evenly across the space. For example, if you are looking at a rural area from a remote sensing image, right, you will probably say that most pixels in this area, they are vegetation, which means this area is homogeneous. There is only one land cover type, vegetation. There is no change from one pixel to another. That is called homogeneous. On the other hand, if you are looking at an urbanized area from a remote sensing image, you will see that those pixels within urbanized areas, they are covered by a lot of land cover types: vegetation, water, asphalt, right? Plastic, metal, because it's urbanized area, it's very complex. So if that is the case, we can call this urbanized area heterogeneous, because the different land cover types, they are not uniformly evenly distributed within the space. Okay, so this is the idea of something being heterogeneous. Okay, so many geographic phenomena can be viewed as spread through heterogeneous landscapes. For example, animal movement through a landscape, right? Urbanization. I mentioned this before, right? And the expansion of urban area, spread of infectious diseases, they can all be considered the spreading of a phenomenon through heterogeneous media. Okay, so that is percolation. That is percolation. So let's talk about the definition of percolation theory. Simulated spread through heterogeneous material modeled as a random lattice. So again, here, we're going to use a lattice to address space, okay, when we're trying to simulate percolation using agent-based models. Okay, and the original problem, it was proposed in 1957. Suppose a large porous rock, which means a rock with a lot of pores inside, is submerged underwater for a long time. Will the water reach the center of the stone? Which means that there is a phenomenon of spreading of what? Spreading of water through a media. What is that? What is that media? The media is this porous rock. Okay. This is the percolation of the water. But here we focus on the media, right? So we focus on the structure of this porous rock. So related problems, you can ask a lot of questions based on this assumed example, right? A porous rock under the water. How far from each other should trees in a forest be planted in order to minimize the spread of fire? This is just like the example we saw here, right? You can consider that each tree is a pore in the rock, and the fire is the water, right? The water is moving from one pore to another pore in the rock, but the fire is trying to spread from one tree to another, right? And how infectious does a strain of flu have to be to create a pandemic, right? This is a very popular topic today, given the severe spreading of COVID-19, right? And how likely is it that information, for example, gossips, rumors, a viral video, will spread widely through a social network? Okay, which means that if you want to be a net popular star, you want to be an internet star, you have to know how to spread your information. You need to know the media, which is the internet, different communities, different internet users. What's the property of this media, and how to use that property to spread your information? Okay, I would say that this is very important today, especially for internet industry, for YouTubers, for streamers. They are being popular not because of some coincidence. They have their own team working for them, working on what? Working on how to spread their information, their images, their videos, their comments efficiently so they can get popular. So that's another application of percolation theory. You need to know the media, okay? And you can also use growth model to simulate the spreading of information itself, okay? So you can say, like I said, a spreading phenomena is everywhere in real world, okay? So now let's talk about different types of percolation models. The first type is very simple. It's called ordinary percolation. There are two types of ordinary percolation, and the first type is called bound percolation. So imagine pouring a liquid on top of a rock of porous material. Will the liquid be able to travel through a path from top to bottom? So here is the example. So let's just assume this square space is inside of that porous material, and each circle here is a location in this material that uh, that liquid or water can occupy. But you should know that this percolation is called bound percolation for a reason. The special part of bound percolation is that you can see some locations they are connected by solid lines, which means there is bond between locations. And for other locations, they are not connected by bonds. What does it mean? It means that if there are closed edges or there are bonds, these bonds allow liquid to flow from one location to another. Okay, and there are open edges. For example, these two locations here, they are not connected by a bond, which means that even one of them is occupied by liquid, by water, that small volume of water or liquid cannot go to the other location because they are not bonded. Okay, so for a given P, P is the probability of closed edges. Okay, probability of finding a closed edge or finding a bond in this space. For a given P, what is the probability there is a path that the liquid can travel through from top to bottom? Okay, this is what we care when we're talking about bound percolation. The primary care, let's say that primary concern is this value P. Okay, so this is a bound percolation, And in this course, we care more about the second type of OP, of ordinary percolation, which is called side percolation. Okay, imagine filling a box with glass and metal balls placed randomly. Is there a spanning cluster, which is a group of metal balls touching each other that reaches from one side of the box to the other? Okay, so be careful here. A group of metal balls that touching each other that reaches from one side to another, which means glass balls that don't counter. Okay, this spanning cluster from one side of the box to another should only be formed by metal balls. You can consider that electricity is trying to travel from one side of the box to another. Obviously, glass cannot conduct electricity, but metal balls can, right? So here is an example of a side percolation. So we can consider all those gray scales, they are metal balls. And all those white cells, they are glass balls. So is there a cluster connecting one side of the box and another side of the box so the electricity can go through? Okay, so the side percolation can be modeled as a random network of vertices and metal balls with probability of P, or you can say proportion of P, right? Glass balls with probability of 1 minus P. Okay, edges in the network correspond to where balls touch each other. So it means that in this space, inside percolation, we use what? We use Van Neumann neighborhood to differentiate different clusters. So what does it mean? So we have 4 small clusters here, painted black. So cluster 1, cluster 2, cluster 3, sorry, 5. Cluster 1, cluster 2, cluster 3, cluster 4, and cluster 5. Why do we consider these 2 small clusters 2 clusters instead of 1? Because they are touching each other, but they do not share an edge. And we're using Van Neumann neighborhood. So these two in this part of the space, we have two small clusters instead of one big cluster. Here is another example. So again, you can consider blue cells as metal balls, and it has a proportion of P, or you can say the probability of finding a blue cell in this space is P, right? And we can say that we have 4 cells marked by different numbers: cell 1, cell 2, cell 3, cell 4, cell 5. So cell 1 obviously belongs to a huge cluster because cell 1 shares an edge with another cell in this cluster. But cell 2, 3, 4, 5, they do not belong to any cluster here because none of them shares an edge with another blue cell. So cell 2 is surrounded by 3 white cells. Similarly for cell 3, similarly for cell 4, similarly for cell 5. Okay, so when we're talking about site percolation, we use Van Neumann neighborhood, which means when you're trying to find out clusters, you have to make sure that all cells in this cluster, they share edges. Okay, they share edges. So some examples, and these examples, they're showing the importance of p-value. Okay, what is p-value? P is the proportion of what? Of metal balls, or you can say the proportion of cells that are occupied by something, by the liquid, by any other thing. So that's the value of P. And P is also can be considered as the probability of finding an occupied cell in this space if you randomly choose a cell. Okay, so probability, P is probability. P of finding an occupied cell. P is also the proportion of occupied cell. Okay, so examples of site percolation on a lattice. So we have 6 lattices here. White cells are unoccupied. Gray cells, they are occupied. And we also have black cells. Black cells are occupied and belong to the largest cluster on the lattice. So let's just compare them. For figure A, the proportion of occupied cells is only 0.2, 20%. And of course, we can say that there are so many small clusters within this space, but the largest one is here. It's very small, but it is already the largest one in this space when p-value is 0.2. And you can say as the p-value increases from 0.2 to 0.4, 5, 5, 6, 8, you can say that, obviously there are more and more gray cells because P is a proportion of gray cells, right? And simultaneously, you can say the size of the largest cluster also increases because the size of black cluster increases. It's very small here, larger, 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 and when P is equal to 0.8, it means that 80% of cells on this lattice, they are occupied. And we have this very huge cluster spreading actually almost everywhere because you can see most gray cells, they are black right now, and they belong to one cluster, the largest cluster here. So you can say the value of P is very important to the spatial pattern of site percolation. Okay, and we'll talk about P later, but now let's just discuss some major questions considered in percolation theory. Okay, what is the average size of a cluster? How many clusters of a given size? Which means if I gave you a specific size, how many clusters have this size or with this size on this lattice. At what value of P do spanning clusters emerge? Here we go. Here we're talking about P. Obviously, the larger the P, the larger the largest cluster of that lattice. But you can see that here we have small, we have largest clusters in A, B, C, and D, but still they're very small. They do not connect one side of the lattice to the other side of the same lattice. So they cannot be considered as spanning cluster. But in figure E and F, obviously the largest clusters in them, they are large enough to connect two sides of the lattice. Actually, four sides, right? You can see this huge black cluster here. It is spreading to 1, 2, 3, 4, all four sides of this lattice. So of course, it's a spanning cluster. It does not only connect two sides of the lattice. It connects all four of them. Naturally, it's the same thing for F, because the largest cluster here in F is even larger than the one from figure E, right? So here we care about the value of P when spanning clusters start to emerge. And also, we also care about another very important parameter here is called percolation threshold. It's called PC. You can consider C as critical, okay? So it's a specific p-value. When large clusters and long-range connectivity first appear at this value, okay? So it's a threshold, which means there is huge change happening at this specific value. When p-value is smaller than pc, it's one spatial pattern. When p-value is equal to or even larger than pc, another totally different spatial pattern will appear. That's the reason why this pc value is called a threshold. Okay, we will talk about this later. And analyzed using network simulation and analytics, it means that these p-values, these questions, they can be answered either through simulation or through pure analytics. Okay, what does it mean? It means that when you ask a question, you want the answer, you can run simulations many times to find many results and try to find a pattern from these results to answer your question. Okay, that means analyzing using simulation. Or you don't do simulation. You use mathematics to describe your phenomenon, and using pure mathematics to find out a result for your question based on that phenomenon. That's called analytic results. Okay, simulation results, they are based on simulations, of course. Analytic results, they are based on pure mathematics. Okay, and also we also care about the flow and spread behavior. How does material flow through the network? And material can be invasive species, disease, ideas, rumors, right? And analyzed using random walks through network. This is not about the media anymore. This is about the material spreading in the media, right? But here in this course, we focus more on the media, okay? And let's talk about that threshold PC. It is the value of P where probability of spanning clusters increases rapidly. Let me emphasize this again. What is a spanning cluster? A spanning cluster should be a cluster that at least connects two sides of your lattice, or two, let's just say two sides, okay? It's more clear, okay? Which means it must be a huge cluster, and being huge is not enough. It must connect at least two sides of your lattice, okay? Based on simulations for a 2D lattice using Van Neumann neighborhood, the PC value is approximately equal to 0.593, which means that if the proportion of occupied cells is around 0.6, then it's very likely to find spanning clusters on this lattice. If we use 2D lattice simulation, but using more neighborhood, obviously it's a larger neighborhood than Van Neumann, right? Then the PC value decreased a little bit to 0.407. And these results, they're based on simulations. And they are also results from pure analytics, pure mathematics. Of course, these values, they are based on different literature, published by different scholars, right? For 2D bond percolation, okay, it's not side percolation. This PC value is equal to 0.5, okay? But again, this is not the focus of the course. We focus on what? Percolation. Side percolation. Okay. This is a table for statistics describing the 6 lattices in figure 5.2. Okay, what is figure 5.2? These 6 figures. Still remember, they are having different values for P, which is the percentage or proportion of occupied cells. Okay, let's get back to this table. Okay, so first column P, right, from 0.2 to 0.8, 6 rows, 6 figures from figure 5.2, right? Number of clusters, you can say that when the p-value is very small, there are so many clusters, and when the p-value increases, the number of clusters decreases because there are more and more big clusters, right? So the number of clusters decreases, and the mean cluster size increases at the beginning, and when the p-value reaches 0.55, the mean cluster size is at its maximum, and then drops, and the probability a cell belongs to a spanning cluster is 0, 0, 0, 0, until the p-value becomes larger than PC value. What is PC value? PC value is 0.593. So you can see here is an abrupt change of P infinite. Probability site belongs to spanning cluster. When p-value is a little bit below the PC value, which is 0.59, right? There is no spanning cluster, which is the meaning of this 0. You cannot find any location, any cell that belongs to a spanning cluster because there is no spanning cluster. But you can see that when the p-value is just a little bit above that critical value, 0.59, suddenly, approximately 47% of cells belong to a spanning cluster or even more spanning clusters. So this huge change happened around that PC value. Still remember what I said before about that critical value? If a specific p-value is below that critical value, you have one spatial pattern. And when your p-value is just above that PC value, you have another spatial pattern. This is what you can tell from figures. But what can you tell from the spatial pattern of clusters? From D to E, right? There is a huge change of spatial pattern in terms of the largest cluster. Indeed, this largest cluster is still small. But when you change p-value from 0.55 to 0.60, it's only a difference of 0.05. But this difference is happening what? Around that PC value, 0.59. So there is a huge change of the in terms of the size of the largest cluster. This is the largest cluster when your p-value is equal to 0.55. This is the largest cluster when your p-value is 0.6. Huge leap from D to E. Okay, so PC value here is pretty important. It's like a baseline when we're talking about percolation. It's a threshold. Okay, so here we have more panels, scatter plots to show different aspects of cluster geometry. First one is the probability of a spanning cluster, occurring, right? So the horizontal axis is for P, right? The vertical axis is P infinite, which means the probability that a site, a pixel, a cell belongs to a spanning cluster. Before, of course, this thoughts they are from many simulations, right? When your p-value is well below the PC value, no pixels, no size belong to spanning clusters because there is no spanning cluster. But when your p-value is above that PC value, this is PC value, right? It's very likely to find the sites belong to a spanning cluster or multiple spanning cluster. Okay. The second panel showing the average cluster size, that is vertical axis against p-value. Okay, you can see that when the p-value increases from 0.5 to PC to 0.59, the average size of clusters increases. But after PC value, the general trend is that the average size of clusters decreases as P increases. Why? Because we have outliers here, right? Some huge spanning clusters with extremely large size. They're outliers. They're pretty big. So sometimes there is a huge value for average size of clusters, but most of them, the average value is pretty low according to multiple simulations. And the third one here is a log-log plot. It's interesting, okay? It's the power law distribution of cluster sizes observed at PC. So at this plot is based on the simulation when P is equal to PC. Okay, so it's a snapshot. And you can see here it's the cluster size S versus number of clusters of size equal to or smaller than S. So it's the number of clusters that have a size smaller than S versus different cluster size S. And here we can see that the horizontal axis is not linear. All those values, they are not linear assigned. Instead of that, it's assigned following log principle. So 10 to the power of 0, 10 to the power of 1, 3, 1, 2, 3, 4. So at each specific measurement, the value is 10 times of the previous measurement. Okay, it's similar for the vertical axis here. It's not from 1, 2, 3, 4, 5, 6, 8, 6, 7, 8, 9, 10. It's 1, 5, 10, 50, 100, 500, 1000. So both axes, they are not linearly measured. Okay, okay, okay. I'm going to show you more information about this power law panel or plot later. Okay, in the next video. Okay, here is the end of the first video of chapter 5, and thank you.